So I joined Ben-Gurion University in 2012 and established the biomedical image computing lab. And we do different projects and in particular develop algorithm and computational tool in order to do analysis. I should solve this. Sorry for the chance. So we do algorithm and tools in order to develop medical image analysis data algorithm. And if you look at these examples, you may notice that we have a particular interest in brain imaging analysis and computational neuroscience, but we do also microscopy imaging analysis and some other stuff. Now, during the years, we use different tools. So, for example, we use level set, variational approaches, graphical models, but recently, like all labs, we moved to switch completely to deep neural networks, and this is what we are doing now. I will split my talk into two parts. So in the first part, I will present our work. Wow. Okay, so I will continue. So I'm splitting my talk into two parts. The first one, as I promised, is lung CT imaging analysis for the analysis of COVID-19 patients. And the other one is graph neural network for cell tracking in microscopy videos. Now, these two talks are very different. It's not only that this is lung CT and the other is microscopy video, but also the focus is really different. I was thinking it might be that the audience is heterogeneous and there are MDs and PhD here. So the focus of the first part of my talk would be more highlighting the differences between clinical measures and imaging finding using deep neural networks, while the focus of the second part of my talk will be more algorithmic, and specifically we use GNN in order to do analysis of microscopy videos. I will start with the first part. I'm sorry for this. I hope now it's okay. Okay, so I will start with COVID-19 severity assessment using deep neural networks. This is a joint work with Professor Ilan Shelef and Dr. Gal Ben-Aryeh, both are clinicians from the Soroka Medical Center. And this work was led by two students in my group, Tal Ben Chaim and Ron Moshe Sofer. I don't need to say too much about COVID-19. We had a hypothesis here. We thought that imaging finding could be related to clinical measures, and we wanted to test that. Our hypothesis is that AI can support, and measure extracted by AI can support clinical decisions, compare follow-up scans, assess pneumonia severity and progress, and predict disease progression. Here you can see X-ray and CT scans, two different patients, and you can see that there is a very significant difference. Obviously, X-ray is much more prevalent than CT, but you can see much more features, and this is why we decided to work with CT images. And here you have an idea on how normal, healthy lung tissue look like with respect to the lung of COVID-19 patients. Specifically, we looked for specific opacities. So we aim to automatically locate, separate, segment, provide volumetric measures of healthy lung tissues, ground glass opacities, here in blue, and consolidation, here in yellow. Now, while you may look really carefully and notice the differences between GGO and consolidation, and in fact, consolidation is much more severe opacity. It indicates worse disease. It's really difficult to tell where is the boundaries between GGO and consolidation, and this is what we aim to do here. We use supervised deep learning approach, deep convolutional neural network. We use training from annotated examples. So we have the input data and we have annotation, the annotation I provided by a clinician. It might worth noting that different conditions would provide different annotation. So there is inter-rater variability. And actually, the different annotation may lead to different paths of training. I will not get into it, but it's worth keeping in mind. Okay, during inference, as usual, network sees unseen data and provide prediction. We use the unit. It was proposed by Ronenberg a few years ago, and we found it very useful. We use 2D data, although our city is volumetric because we only have single annotated slices. And by doing so, we address the challenges. Now, these challenges are not specific to COVID-19 analysis. It's general to every analysis related to medical imaging. We have a small amount of annotated public data. So in fact, we only had 100 annotated slices that we could use for training. In fact, we used just part of them. And then we have volumetric data, but because of the strong correlation between consecutive slices, we couldn't use them. So we use them only for test. There is variability, as you may imagine. There is variability between patients. And there is variability between data sets. Now, usually, different data sets come with different annotation of different annotator, and this should be added to the variability. Another problem is class imbalance. So, for example, in this case, most of the images are background, 25% are lung tissue, healthy lung tissue, and the rest are consolidation and GGO, and this may cause some difficulties. Another difficulty that I mentioned before is to outline the boundaries between GGO and consolidation, which is really difficult even for a human annotator. Few more words about our data set. So we have 100 CT slices and training test split is 74-26. We have a notation of two different raters. So one comes with the public data and the other is Gal, one of the co-author of the paper. We have volume that we keep them only for test, altogether in a makeup of 790 slices. Again, we have a notation from two different raters. And we have our own private data set provided by the Soroka Medical Center. We only have 23 volumes. In fact, we started with 100 volumes, but it turns out this is how medical data from the wild look like, a lot of duplication, post-COVID-19 patient scans, motion artifacts, so on and so forth. So eventually we ended up only with 23 volumes, and this data is not neatly arranged as you may expect in challenges. And for each patient, what we have is a clinical risk score that was calculated based on demographic and clinical measure. For this data set, we had only single annotated slides for each of the volume. So our method is very standard. We use all the tools we knew in order to get the best of the data. So we use a hierarchical segmentation network, and we find it very effective to accommodate problems where we have class imbalance. We use transfer learning, and we found it very effective in order to overcome the lack of training data. So we use VGG16 as a transfer learning. We use weighted loss function, again, accommodating imbalance, and an ensemble of M networks that actually helps us to provide better results. It also helps us in our generalization capability. Another aspect of the work is segmentation uncertainty and clinical risk analysis. Loss function, so the network, the hierarchical network compound was trained end-to-end. We use binary cross-entropy for the first part of the network compound, which is the LangNet, LNet, and we use categorical cross-entropy for the CNet. Altogether, there was an interplay between the two networks. They were trained end-to-end, but they were the algorithm frees part of the network at a time. We also use the dice loss and regularization, but these are just technical details. So what can the ensemble provide us? We have prediction for background, for GGO, for consolidation and healthy lung tissues from all the ensemble networks. Now, if we use the soft output of each of the networks and then we sum together, we can get a measure of uncertainty, and this is what we did. So if we combine the GGO and soft segmentation, we have infection certainty map, and we use that. But why is it good for? Why do we need an infection certainty map? So I had an hypothesis that the disagreement between the networks in the ensemble might be correlated to the disagreement between raters. And we actually checked that. So we have two different raters, rater 1 and rater 2. And here you can see an example of annotation in light blue and in yellow. So let's say these are annotators. In fact, they were both men, but I'm committed to gender parity, so I put a female annotator here. We can look at the binary segmentation, and if I do the intersection, you can see that there is a significant overlap, but still there are differences. So the main idea was to compare the intersection and differences to what we get from the ensemble. But before we do that, let's have a look at some visual segmentation results. So each row is a different, each row is a different case. Each column, so the first column is the input, second column segmentation by rater 1, and third column is the proposed segmentation. And you can see that there is similarity. Again, light blue is GGO, yellow is consolidation, and healthy lung in red. Now, you can see the certainty map and compare it to inter-rater variability, and visually it's look nice. The question is, what about quantitative measure? So we did it. We actually took the ensemble soft maps and calculated the entropy, compare it with the dice loss. So the dice loss was easier to compare between two different raters, and you can see that these two are correlate. Quantitative result related to the dice score. So we compare ourselves to different algorithms. So for example, the InfNet. The InfNet was not tested, or the Radiopedia dataset. I would like to remind you that the MedSeg dataset was used for training and test. It was splitted, while the Radiopedia was used, the volume was used only for testing. So we outperformed the InfNet. We actually outperformed the UNet, which is baseline. With the NNUNet, well, we had a problem. Because the NNUNet actually win in all the medical challenges. So the key idea is as following. It has training data, and it is also trained on the hyperparameters. However, as you can see, it is very bad in generalization. Okay, and to compare and to see if our results are good enough, we compare the intersection of the first rater with the second one, and you can see that, well, our proposed algorithm provide better result, better dice segmentation result. We did the same for consolidation and for binary. I will keep it quick and just mention a Kaggle competition where we won the second place. There was a time it was really popular to check yourself on COVID-19 segmentation in lung CT. Another another measurement we can provide is the volumetric measure. We look at the segmentation, simply integrate, and you can see that for GGO and CON, for the data set tested, we're doing quite well comparing with rater 1 here in green and rater 2 here in blue. The proposed one is in orange. Now I would like to move to the most interesting part, which is our public data set, the one that was collected by Smoka. And we did a volumetric. Now we can't test that. We only have a single annotated slide from each volume. The dice was reasonable, but we couldn't say anything about the volumetric quality of our segmentation. So what we did, we decided to compare it to the clinical measure. Before that, we calculated two different measures. One we call the extent of the disease. It is the sum of the GGO and the consolidation and their ratio with the lung, healthy lung tissue. And the other is the severity consolidation with respect to the sum of the GGO and consolidation. And remember that consolidation is a more severe appearance of the disease. In order to calculate with clinical parameters, we calculated the clinical risk score. And one of my students decided to demonstrate it by providing to you an hypothetical patient. Her name is Sarah. She's 65 years old, and some of her values, clinical measures, are below or above or within the normal range, and her clinical risk score is very high. It is 12. You simply sum the number here. Now, we did this calculation of clinical risk score, which means, whether the patient would need ECMO or extra oxygen or intensive care or whatever. This is something clinically done in routine, and we compare it to the volumetric prediction that we got from the imaging. Now, this is the result we received, limited data set, 23 patients. It may not be so exciting, but remember that not all the clinical and demographic values, for example, age, not necessarily related to the infection in the disease. Still, there is some correlation. I can also tell you that since we went over all these patients, and I can tell you that there is one young patient who had a very severe lung disease, but it was young, and all these clinical measures were good. On the other end, there was an old female, and she, her lungs were completely clean, but she had diabetes, and she suffered from many other problems, and she was old, and she got very high clinical risk score, while her lungs were relatively clean. So I think this might be a good starting point. Obviously, we need to continue working on it in order to get something. So I would like to conclude this, and let me just change this argument into questions. So do we need multi-class segmentation in COVID-19? Would that provide additional information? What can we say about generalization capability, the differences between the data set? Does certainty map actually correlate with the consensus between the raters, and volumetric prediction versus clinical risk assessment? So before I conclude this talk, I would like to acknowledge the MOST, Ministry of Science and Technology and Space, and two clinicians that actually help us, Israel and Amit, for calculating the SUMC clinical measure. Microscopy imaging analysis is a very important tool in system biology research. Our clinical collaborators study cancer research, do cancer research, for example, and they study single-cell features such as protein level, cell-to-cell interaction, dynamics, proliferation rate, so forth, and so on. Now, in order to do that, you need to do single-cell analysis, and this is a very challenging problem, specifically since the data varies and the existing algorithms are not sufficient, and manual annotation is impossible in many cases. Okay, so two important aspects in single-cell analysis are annotation or segmentation and association of cells instances in consecutive frame into a single biological cells. So the end result is the lineage analysis, as you can see here, and this can provide a very complex measure, such as inheritance, dynamic, cell path, and so on, that our collaborators, our biologist collaborator can utilize in order to understand. However, so in this work, I will concentrate on cell tracking. I will assume segmentation is given or annotation is given, and there are many challenges. The first one is that it apparently seems that the cells are rapidly moving. In fact, it all depends on the sampling rate, the dynamic sampling rate. So it turns out that when you take photos with a microscope, they are toxic. This is why biologists try to have a photograph every 15 or 20 minutes, and if the dynamic of the cell is higher, they appear to be moving really rapidly. Another thing which is very different from car tracking, for example, is that their motion appear to be really random. Another issue, which does not happen to car, cell splits. They change their shape. Actually, the fact that cell splits cause another issue because you start a sequence with like dozens of cells, and then it turns to be hundreds and thousands of cells at the end of the sequence, and this is a real problem. So as you can see here, the boundaries are not well defined, and there are other issues, and specifically, if you look at different sequences, they look completely different. There are two main approaches in order to do cell tracking. The one is the local one. So you simply look at consecutive frames and you calculate the association between cell instances in pairs of frames. The other approach is the global approach. That is, you look at the entire sequence. In this case, you may present the sequence in a graph, and classical algorithm tends to solve this problem using combinatorial approaches. Nowadays, we can split our related work slide into, in a two-dimensional way, into classical method and deep learning method, and there are already local cell tracking approaches that use deep learning. We are targeting here a global deep learning approach that uses deep learning. So in order to do that, we cannot use convolutional deep learning network. So the cell data does not appear in a grid. It's unlike video or images. So instead, we use graph neural network, and I can think of different data sets that have this form. There are proteins, social networks, and so on. And to our purpose, we use cell data sets. Now, main issues processing data represented by graphs is first, learning the mapping, and second one is aggregation function. Our approach consists of global approach, deep learning. We use graph representation. We use graph neural network. And finally, we use message passing. Now, in order to formulate a problem using graphs, let's consider 3 consecutive frames. Got complicated here. So we define a graph by G. We have vertices, so each cell instance is a vertex in the graph, and we have edges, and these are the potential connections. So these are not necessarily the final connection, but the potential ones, and we assume there are connections only between consecutive frames, and they have a very well-defined direction. Just to give an overview on the entire method before we move on. So we have an entire sequence as our input. We use deep metric learning in order to learn the features. We use graph neural network in order to do the calculation or the optimization. And then we solve for possible paths in a graph, that is association of cell instances belonging to the same biological cell, simply by a binary classification. So at the end of the process, we need to say which edges are active or not. We remove from the graph the non-active. The length of the sequence? No, we do not sample. We take the entire sequence. Yeah. So we assume that we have a notation. So what we did, we apply a segmentation algorithm. We also have a segmentation algorithm. Alternatively, we use a notation algorithm in one of the cases where we didn't have a ground truth. But it's necessary, otherwise we couldn't. Yeah, it's a good question. So we assume our data is annotated, and we use this annotation in order to provide the tracking. Yes, a good question. So this is the entire change. And let me just now concentrate on different parts. So our input is sequence of frame, grayscale images. We preserve cell instances. The dimensions are either 2D or 3D. Actually, it's better to work with 3D. We got better result in 3D because cells do not hide each other. They exist in the 3D. The corresponding sequence of label maps is necessary, as I mentioned. So we either have the boundary of the cells or we have marker that indicate the cell centers. Okay, so let's have a look now on the embedding. So graph representation is defined by X, Z, and E. These are 3 matrices. X is actually the node feature matrix. And you can see it has the size of V and the dimension of the number of features, the node features. The same applies to the edge features. It has the dimension of the edge or the cardinality of the edge and the size of the feature vector. And E is the edge connectivity matrix. It is 2 by E, and it represents all possible links of cell indices. Now, this representation is much more economical than affinity or adjacency matrix. L is the step number, and we aim to predict y hat. And what is y hat? Y hat is a binary matrix which indicates which edges are active or not. So eventually, we solve a simple binary classification matrix for the entire sequence. Another important aspect is the message passing. So have a look at the central vertex here or cell instance here, and look at all the incoming edges from their neighboring cells. Okay? They would influence the cell features, all the incoming. So they are first-degree neighbors, and they would influence. So the first GNN block would be a first-order influence on each of the cells. So just to demonstrate that, here you can see again an illustration, 3 consecutive frames, and this is the first block. So we can see that one frame influenced the other. But if we add a second block, so we have a second form of interaction. So this would interact this. So T would interact T plus 1. T minus 1 would interact T. But then, after applying the second block, we have second-order neighborhood. So if you had more and more blocks, our message passing extend, and actually we use 6 blocks in order to do that. Okay, one important aspect that we added to our graph neural network is that we have edge encoder. So the focus is the edges and not the nodes. Okay, so we calculate node features and edge features consecutively. Okay, so in fact, each node or each vertex is infected by all its nearby neighbors. And if a neighbor has a strong influence, it would affect the feature more, and vice versa. And now for the edges, each edge is affected by the connected node. Okay, and more specifically, we define a DS block. A DS block looks like this. So this is simply the differences between 2 consecutive features. And then we have the cosine in order to define the interaction, the full interaction between them. And in the same manner, we have edge feature matrix. Okay, so the reason kind of interplay, we influence edge features, and edge feature influence node feature, and node feature influence edge features, and it seems that the message is passing throughout the graph for the 6 blocks until the 6 related neighborhood, until we change the feature, and then we can do classification. So we can use a simple classifier in order to classify the edges. What a classifier does, simply remove the non-active edges. How do we solve for mitosis? So this is kind of post-processing in many cases. In some cases, one instance is connected to 2 or more instances in the next frame, but not all the time. So for that, we need a triplet. We define a sequence by the initial cell instance, the final cell instance, and if 2 sequences that are neighboring end at the same time, and if we look at the next sequence, it starts when they are ending. So, sorry, the other way around. If 2 starts when the other one ends, then we can say that the mother cell is this one. Now we come up to the deep metric learning. So how can we define the features? So we have spatiotemporal features at the center of the cells, the volume, and so on, but we wanted to select features that would discriminate cell instances that are from different biological cells and would put together cell instances that belong to the same biological cell. And we did hard mining and deep network learning in order to do so. I will not get into too many details here. Maybe I would just give an illustration. So say this is an anchor cell. I can find out the most related instances, and I can do it again and again. Okay, until I build 3 sequences. I have another illustration of hard negative and hard positive. So for example, let's look at hard positive. So in red are the negatives, and if a negative is closer to the anchor than a positive, then it's hard negative and vice versa. So we do deep metric learning using this idea. We add to these spatiotemporal features. And we use both spatiotemporal features and deep metric learning feature in order to build a vector of features for each edge and node. Now, what about our loss function? The system is trained end-to-end. So we have a weighted binary cross entropy because there is some imbalance, okay? There are more non-active edges than active edges. And we have the deep metric learning loss experiment. So we did two types of experiment on two different data sets. One is the C2, C12. It is a private data set that we got. It's an open, but it's now open, but it used to be private. And we applied our network to different sequences of the same type of cells. Here you can see some qualitative tracking results, so the lineage analysis. In addition, we did quantitative tracking measurements using association accuracy and target effectiveness measures. So the target effectiveness measure is quite tricky because we actually calculate the ratio of the number of frames in a track that were not set correctly. And this is really difficult, in particular when you have hundreds of frames. Okay, so let's do some comparison. And here you could see some comparison. So we compare two different methods. So we compare different data set, the control, and different data set with different growth factor, and also look at the average. You can see that our method in the last row outperform all other methods. And let me just have a specific look. So the first two methods are global method, but done in a classic manner. The next method that we compare to are deep learning method, but are local ones. And in both cases, our algorithm provide better results. Now have a look at the last rows. So we actually change the dynamic by sampling every fifth frame. And even in this case, our algorithm overcome and outperform the other competing algorithm. So this simulates much higher dynamic, 5 times the higher dynamic. Another experiment that we did is that we used the cell tracking challenge data set. So this is a very fair and balanced competition because you can see the ground truth. You actually give your result to the organizer, and they calculate for you the scores. And then you can compare your scores to the others, and everything is publicly published. Here you can see some example of the sequence, and here you can see our scores for different data sets. So as you can see, these data sets are of different dimensions, either 2D and 3D, different cell types, different acquisitions. So the first one is phase contrast and the other is fluorescent. Different number of frames and different resolution. And here you can see the score. So for 3 data sets, we won the first place, for one, the second one, and for 1 data set, the fourth one. We did some experiment, an ablation study, with and without spatiotemporal features, deep metric learning features, edge encoder, different structure of graph blocks and for the different quality measure. And in all, our algorithm outperformed the others. So just to sum up, what we did here, we did a self-tracking algorithm that simultaneously provide all the paths in a sequence, okay? Not in a local, in a greedy suboptimal manner, but all together. It is solved as a simple binary classification task, active and non-active edges, and we use graph neural network, and I think it's the first time graph neural network was applied to such data for such tasks. That's it. Just before I conclude, I apologize for this. I would like to acknowledge the BSF, the MOS, and the code, and actually the code of the COVID-19, and also this code is available at GitHub, and you can contact me with any questions.